Hey everyone, this episode is going out on Wednesday, the 11th of October 2023, and I just want to talk about our small group sessions, coaching sessions, which are starting again on this Monday, the 16th of October 2023. It's 8 90-minute sessions running Mondays at 7 p.m. UK time, worldwide on Zoom. Going right through to Monday, the 4th of December this year. So what do you get in the small group sessions? You get cutting edge science explained clearly and in simple terms. You get a discussion of subjects that affect your metabolic mental health, including food addiction, dietary modes like keto and carnivore, why it works, how to get your macros right, gut health, getting enough fat, supplements, and you also get a non-judgmental and totally confidential space to talk about what matters for your mental health. With experienced practitioners, myself and senior psychiatrist, Dr. Rachel Brown, who understand how powerful diet and lifestyle can be. So what have other people said about doing this small group session with us? Well, Martina said, great, it's geared at just where I was at and just what I wanted to know. Ali and Rachel's knowledge and commitment to metabolic psychiatry and their desire to share this was an absolute gift. Elizabeth said, go for it. I can't believe the difference it made. I still have more to problem solve, and it's great to still have the forum and possible subscription, but finding freedom from binging has been a dream come true, and many times where the clouds of depression parted has been so good. And Chris said, I would actually say this is an exciting and cutting edge therapy. It's something I wish I'd known about 30 years ago. We won't be recruiting again for small group coaching until February 2024. So if you've wanted to work with myself and Dr. Rachel Brown and you feel like the group sessions are the way to do it, you have to act now because they start on Monday at 7 p.m. UK time, worldwide on Zoom. In previous groups, we've had people from America and elsewhere. So if you can make 7 p.m. every UK time, which is about midday to 2 p.m. ish on a Monday in America, every Monday from next Monday, the 16th of October until Monday, the 4th of December, and just go into Christmas time feeling great. Then head over to bit.ly/MetSci Group, METPSY Group. I'll put it in the show notes as well. If you're on the fence about it and you're not sure about the process, then you can have a free 30-minute chat with me, either about the group sessions or going one-to-one, and that's at bit.ly/Ally Transforms, ALLY Transforms. I'll put that below in the show notes too. So please head over to bit.ly/MetSci Group, book yourself a place. There's 20% off now, and it's a great deal. So please enjoy today's episode with Andrew and with Isabella, and I'll see you soon. And we're recording. And I'm delighted to have with me Andrew Scarborough, who's a freelance writer and editor focusing on brain cancer research, and Isabella Cooper, who's a doctoral researcher at the University of Westminster. Welcome to you both.
Thank you for having us.
Thank you.
So, Andrew, you've been on the podcast a couple of times before, and you've embarked on some research recently, and it is really that's brought you on again for a third time. So do one or both of you want to just kick off and say, what is the research about? Maybe even just a wee bit about yourselves if people haven't heard your stories before, and then we can chat about it.
Yeah, so over the past couple of years, I've been working as a writer, as you mentioned, medical writer, and an editor, mainly for the International Brain Tumor Alliance, but also for other organizations, big and small. And I wanted to get back into research. So I decided to tag on to progress on some of the work that Isabella has been doing on metabolic phenotypes for healthy people, and those on ketogenic diets, and then to progress that onto brain cancer and to establish a metabolic phenotype for high-grade glioma, newly diagnosed high-grade glioma brain tumors.
Brilliant. And can you break down for people why thinking about metabolic phenotypes is worthwhile, one or both of you?
So with regards to obviously what we look at in ketogenic diets is essentially that there's a big role of glucose and insulin in pathology. And, you know, it's quite well known that with diabetes or obesity, which are conditions of dysregulated glucose metabolism and therefore dysregulated insulin metabolism, those two conditions actually increase the risk of chronic diseases, one of the main ones being cancer, cardiovascular disease, Alzheimer's, and they all have some overlap and risk factors for each other. And so, you know, metabolic syndrome essentially would be a metabolic phenotype. So a package of how people present, and not just visually, but in terms of their blood markers, you know, and like most things, it's on a spectrum. You can be just newly diagnosed diabetic or very, very diabetic, and that would be a spectrum there. And you have people in the normative ranges who don't present with dysregulated glucose, and they may have normal, healthy insulin, or actually have perniciously higher insulin that's keeping that glucose, suppressing it and keeping it looking good. So this would be pernicious, and essentially they're sort of on their way in this phenotype spectrum, on their way into the hyperinsulinemia and then hypoglycemia spectrum. So the normative population we class as phenotype 2. So they would have normal glucose. They even would have what appears to be normal insulin, but be on a spectrum. And then, you know, as they go up on that spectrum, once the insulin is much, much higher, at some point the body's saying it's not going to respond to that insulin. It's going to need more insulin. And then eventually the glucose is actually not able to be controlled either. And then you're into phenotype 3, and you're progressing in stages there where the glucose starts to just always be higher with higher levels of insulin. Now you've got what's classically known as type 2 diabetes or pre-diabetes. And the reason why the normative population we say is phenotype 2 is because there's actually a phenotype 1, which is people who have chosen to live in a particular lifestyle that keeps their insulin so low, but in a healthy low, not a type 1 diabetes low. It's not to do with the fact that they can't produce insulin, but their lifestyle keeps it very low. And that's a combination of restricting dietary carbohydrates to a degree that is within a healthy level for that individual person. And there's a great degree of variability in that. Through to fasting, time-restricted eating, 1 meal a day eating, as well as exercise on top, sleeping well, all these sort of things that are going to affect blood glucose, whether it's from dietary or lifestyle practices, that's going to consequently affect insulin secretion. And those people would have a background amount of ketones, whether it's sort of low grade, 0.3, 0.4, nutritional levels, 0.5 and above, or therapeutic levels, which is half of your glucose levels, if not, let's say, you know, between 3 and 5 millimolars of ketones. So there's those sort of degrees there. And this is what we call phenotype 1, which is more akin to sort of evolutionary man, we say, evolutionary humans who, you know, go back 300, 400 years. They don't have 3 meals a day. They don't have refrigerators, so they can't be storing perishable foods like bread and things like that, or fruits are not readily available. You know, so no 3 meals a day. Fruits and starchy foods are seasonal, less food available in the winter, those sort of things, more physically active lifestyles. And so assuming that our health is optimized based on the evolutionary pressures that we faced, then this phenotype 1 would be more akin to what's likely a healthier phenotype for us, especially if we were just to model that in whole animal studies where first and foremost, animal studies are in caloric restriction, which effectively puts you into ketosis, some degree of ketosis. And in those kind of studies, animals have lived not just longer, but lived healthier, without disease, functionally healthier. And then in those kind of animal models, they've added ketones to them, or they've had ketones without the caloric restriction and produced not only the same results, but better results. So, ketones are a very good marker in the absence of type 1 diabetes. Ketones are a very good marker that your insulin is below a level that is pathological for you, because it's insulin that ultimately suppresses your body's ability to produce ketones. So once we know that kind of spectrum there, and when we know that hyperinsulinemia conditions, obesity, type 2 diabetes being the obvious ones, although it's not always just these two, because you have lean people, about 40% of lean people who are metabolically dysregulated. But anyway, we know that they have higher risk of cancer. So there's a metabolic dysregulation. It's also established that in cancer, it's considered sort of there's a reprogramming of metabolism. So it's sort of thought of the DNA is reprogrammed. The cancer cells have reprogrammed the metabolism of the cells, the tumor. Now, there's a lot of argument and debate on that, and each of us have our own opinions depending on what literature we have read. But irrespective, it can be agreed there is a difference on the metabolism, on how cancer cells fuel themselves. So we know there is that difference. So, some people want to go down the line of looking at genetic changes, receptor expression changes. That's fair enough. We've got lots of people who like to do this research, so that's great. On the other hand, I think there's a very, very strong commonality amongst tumors is their dysregulated energy metabolism. And, it's pretty much, a safe bet, one could say, is if you turn off the tap, they're going to struggle. And if you can turn off the tap for their fueling sources, whilst enabling the energy for your healthy cells, this is obviously going to advantage your whole body, so the whole organism, in order to fight tumors. And, at the end of the day, you need to have a healthy system. And what you find in cancer patients is the whole body is not well. There's a lot of other stuff going on. And that environment, not just the microenvironment, but that global environment in the body is actually enabling that tumor, very much so. And I think the biggest struggle, and Andrew knows this very well through not only himself, but also through his consulting and advising many other people, is that a lot of patients have really struggled with oncologists to get them on board to look at their metabolism, to run the necessary tests. And when you hear doctors say, oh, it's nothing to do with it, or there's no data on it, there's no literature on it, it's like, well, it's not there because you're not looking for it, because you're not testing for it. And as we all know, when it comes to disease, the earlier you catch it, the better a chance you have. And this would save the NHS so much money if they were testing basic metabolic parameters, because they would be catching things earlier. And I'm not saying, therefore, don't go down the standard of care route, but what it could be is implementing nutritional and lifestyle processes could actually facilitate sensitivity to drug treatments or other treatments that will actually enhance their efficacy whilst decreasing the negative downside toxicities. And so anyway, coming back to our research, Andrew and I have known each other for quite some time, actually. We've studied together in our undergraduate degrees. And I had sort of quite a passion from then, and then it progressed into my PhD research to say, well, you basically always hear from the medical establishment, well, ketogenic diets are not really, they're not, it's not about just not sustainable. They're not actually good to do long term, and they're not for healthy people. And so I just sort of thought, well, first, I know that's not true, because I've got a bunch of friends who have been in ketosis forever, like really for such a long time, and they've never had any diseases, and we're all doing really well, including myself. So I thought, well, that's not true. So that's a bit of a problem, isn't it? So I decided, well, if there's no one else that's going to do it, I'm going to do it. I'm going to do the research and get that data from healthy, long-standing, talking, a couple of years and above, therapeutic levels of ketosis that they sustain purely because they like it. Like, that's it, and I'm going to get that data on them so we can show, well, it's clearly okay for regular people. No downsides seem to be coming up. We measure 90 biological markers as well as anthropometrics and, RQ and all the usual sort of stuff. So we measure all of that. And that's been progressing, and it will complete by the middle of February next year. So it's very exciting. And in that time, over the course of some time, Andrew and I were talking a lot and saying, well, look, wouldn't it be just hugely beneficial to phenotype at first brain cancer patients, because they really do suffer such a tight window of being able to do something and turn things around. And so we got talking lots and lots and lots, and we said, okay, yeah, let's figure out how we can do this. Let's get a project going that we can phenotype and see where brain cancer patients sit on this metabolic phenotype spectrum. At least give them that data so that when their oncologist says there's nothing there, or what are you talking about, they could have a paper and say, look here, these group of brain cancer patients, their blood markers were tested, and look, it's different, and hopefully give them that.
Crucially as well, you're able to see what happens from as soon as they're diagnosed through to treatment, whatever interventions they're having, whether that be diet related or repurposed drugs, anything. So there's no, in terms of the inclusion criteria, we're not being picky about what people are doing, but we're taking a note of it. And crucially also pairing that with the imaging and considerations of any kind of medications they're on, because that will be making a difference as well.
Very interesting. Yeah. It's always so tough to communicate any of the powerful points that you made there, Isabella, to people who've never heard about this sort of thing before. And so you end up starting off with this this statement that a great majority of the Western world, at the very least, but probably most of the world, are metabolically dysregulated, that phenotype 2 at least, maybe 3, and that until, generously speaking, 15,000 years ago, really pretty much everyone was highly likely to be living in phenotype 1. You can tell from exploration in the last few hundred years that humans are ingenious enough to live on a variety of diets in a variety of settings and so on, but that it was all phenotype 1. And that it's really if you can get them through that and on board, and then say, well, what is it that really identifies good health? This phenotyping and this biological marker testing can be an extremely convincing way to talk to people about it, because there's a lot of kind of loosey-goosey stuff that goes on about just feeling better in yourself, and that could be true of Slimming World or Weight Watchers or whatever. Also, there's a lot of misinformation about needing to be under 20 grams of carbs a day, or else you have to leave this Facebook group because we all hate you. And there's sometimes not much room for the kind of nuance that there is. When I was talking to Ian Campbell on the podcast recently, he was saying that CSF lactate is a really good metabolic marker of bipolar, and to have that as a patient talking to your psychiatrist is so powerful. To link specific metabolic processes to a disease is really powerful. And so would you say that that's the main thrust of what you're doing, or is there other angles to it?
You mean in terms of like profiling those blood markers and
or cancer specifically?
Yeah. So in the first study, the metabolic phenotypes in phenotype 1 individuals, obviously we're not phenotyping cancer because these participants are super healthy. What we're developing in that is you can say a library or database where we can see what their signatures are. And
gives us something nice to compare against.
Yeah.
And also to then see from that individual data where the shift is. And we can even pair that with the genetic mutations that we see. So we don't have to necessarily throw that out the way. It's useful.
No.
It's useful to see how that may be affecting the patients metabolically. And also, if we have the opportunity to even compare it to, there's a type of scan that they do at Charing Cross Hospital called an MR, it's MR spectroscopy. So it's a type of MRI that allows you to see the bioenergetics of the tumor area. It can also diagnose certain types of epilepsy and where they're coming from. But in terms of cancer, these brain tumors, it's allowing you to identify what type of tumor you're looking at and what fuels it's taking up primarily. So you're able to see the biochemical changes even over time, over the course of disease. I've actually had that scan. It was really interesting because it showed that my tumor was highly glycolytic. And to me, that was actually quite a surprise because you usually see that in lower grade brain tumors. And also because mine was an IDH1 wild type tumor, which just means it's metabolically, in terms of how you pair that up with the metabolism, it's metabolically different in that it's more likely to take up glutamine for energy as well as the glucose. Glucose will always be a primary fuel, whether you're saying, okay, there's these other fuels that the tumor's taking up. But in this case, what we're doing, what Isabella and I are doing is we're looking at all of these metabolic markers and then identifying patterns for maybe different conditions. It gives us lots of discussion points to look at, and it allows us to see how the different stages in terms of treatment, no treatment, whatever interventions happen, if there's a pseudo progression where it looks like the tumor's getting worse, but maybe there's some necrotic tissue or inflammation, something's happening, or if there's even pseudo regression where it looks like from radiotherapy, it looks like the tumor is regressing, but then it starts to quickly get bigger. That often happens on, there's a drug called Avastin, which is an anti-angiogenic drug that's often prescribed when everything's failing. And it's quite difficult to get, but people push for it for some reason because it has this pseudo regression where it's or pseudo response where the tumor starts to shrink and then it seems to spread everywhere. So it's trying to restrict the blood vessels from growing and expanding to provide nutrients for the tumor, but actually it's not selective really. So you're then having this actual spread. So it improves progression-free survival, but overall survival isn't changed. And so it would be really interesting to see if patients are on Avastin, what's happening metabolically from that.
Yeah, absolutely. I mean, this is something that's come up a few times on the podcast. Cancer is not a disease, it's a kind of umbrella term for a particular type of thing going wrong in the body that is tissue specific, and each one is peculiar. And so getting as much data as possible on any of them is clearly good ammunition. And it's also individual to the person, isn't it? Because it's happening in their body. And so just like someone who is in phenotype 2 or 3 might end up with Alzheimer's rather than cancer, or both, or neither, or something else, because of their genes, then a cancer in person A might be different from that cancer in person B, but the more information you have, the better armed you are, right?
Yeah, I also feel as though the brain is unique because it takes up 20% of all of our metabolic energy. So yeah, for many reasons, I think that this is a very interesting piece of research, which can, in the next stage, identify what interventions we could use and what would be most useful for different people within that.
Yeah, and that's the sort of thing where, yes, every single person, when we say they're different, it's in the sense that we had a slightly different childhood. We had different medications we were exposed to, different dietary styles exposed to. Even our mothers, when they were pregnant with us, had different medications and different food types and things like that. And those things are going to have long-term, an impact in how we progress in our life. So, our epigenetic regulation, our microbiome, all these sort of things. And so that does result in our own unique thumbprint, quite literally. But we can say very confidently that majority of cancers are highly glycolytic. Now, that doesn't mean that you're going to have a high fasting blood glucose, because of course, if you've got cancer cells that are guzzling up glucose, they're likely to keep your glucose levels actually looking pretty good. So this is most likely why a lot of oncologists or researchers are saying, yeah, but hyperglycemia or type 2 diabetes has nothing to do with cancer, because you don't see dysregulated elevated glucose. That's because you've got dysregulated tissue guzzling all that up. And as a result, when you have normative levels of glucose, you actually won't see very high levels of insulin, not normally. But in the bell curve of the reference range, it'll be in the upper half in terms of the amount. And again, the reason why the reason why is more so that that insulin level is quite unique for each person, their sensitivity to carbohydrates and things like that. And so you will find normal, healthy, non-disease people, their insulin levels will be in slap bang in the middle of that normative range. And you will think that, you're perfectly healthy. But the only way of really knowing if you're really healthy is actually whether you're sustaining ketosis 24 hours, not just in your morning ketones, but through the day. And this is a way to understand if insulin is actually not good for you, because the reference range, remember that reference range is for a whole population. It doesn't really tell you what's really good for you. And so that's really important with regards to cancers and brain cancer even, because insulin is the master hormonal regulator. It governs all the other ones. It governs the sensitivity to all the other ones. And so, when you have cancers that talk about, hormones and estrogen and things like that, well, the cell sensitivity to those growth factors is actually governed by insulin. The same like vascular angiogenesis, things like this is actually really governed by insulin. It's so you can have just a minutely elevated insulin that's then subsequently going to affect hepatic production of binding proteins that bind to hormones or not. So insulin will suppress those binding proteins, making those hormones much more bioavailable, as well as affecting the production of the hormones to increase them, as well as increasing the receptors on the cells, and as well as increasing the intracellular part of the receptors to allow them to amplify, to magnify that signal intracellularly. So multiple layers that insulin is doing that. And so that's why we need to phenotype these sort of things, because, as we've said, looking at genotypes, so your genetic profile is definitely valuable. Looking at your epigenetics, I think, should be done a lot more. And then metabolic phenotyping is equally, if not more important, if you ask me, because it's actually it's one of the few things that's most common between people with cancers, because that's the whole body something going wrong, essentially dysregulated. And if it's not presenting with cardiovascular disease, not presenting with Alzheimer's disease, it's likely presenting with cancer. And it's just, in different tissue types around the body. And if you have to look at cancer, you don't hear about cancers of the bicep. You know, you don't hear about cancers of the heart. The cancers that you really hear about are in cells that have a natural high proliferation. So it's a high turnover. So these are cells that actually, you know, they split and divide naturally every 3 days, 7 days, you know, up to a month, let's say. So they have a high turnover. So that means their genetic profile is to actually divide and replicate on a regular basis, on a short-term regular basis. So that's a combination of all your cells have the same genes, but they are regulated. And that regulation is the epigenetics, it's what's on top regulating. And so depending on how it's regulated, it expresses as a heart cell, you know, cardiomyocyte, you know, expresses as hepatocytes, liver, or brain. So that's one area of how it's regulated. Another on how it's regulated is how much it gets to divide, you know, grow and divide, grow and divide. So you get some cells that are terminally differentiated. That would be neurons in your brain. So those aren't going to divide again. That's it, it's done. That's why you don't really hear so much about a cancer of neurons. When we talk about brain cancer, it's actually the glial cells. So those cells do grow and divide. And essentially, if you look at all of these cancers in different tissues, their epigenetic regulation, that you see that they grow and divide naturally, that's part of their life cycle. The pattern of their epigenetic regulation is much closer to embryogenic epigenetics, so embryonic cells, because obviously embryonic cells have to divide, you know, divide enough to make a baby. And so that's the epigenetic regulation there. And they are under a much more hypoxic situation at some point before you have the placenta and the umbilical cord fully developing. They're under a much more hypoxic condition. And so they have that same kind of exposure of lower degree of oxygen and therefore burning up glucose. And in actual fact, you look at healthy cells that must grow and divide, they actually trigger all the hypoxic pathways that enables that division process. And so it's the same process that you see that is happening in cancer cells, but not stopping. It doesn't have the, okay, we need to stop that now. And so understanding that it's actually part of normal physiology. It's not just pathophysiology. And so in the case of us phenotyping this, you know, the aim is for people to be given those tools in terms of, you know, restricting that hyperexpression of insulin, which can be done dietarily effectively. That's going to empower them, give them confidence. I mean, I've had so many people who, you know, are undergoing care, and there's a nervousness, a fear to not follow what they're, and I'm not saying don't follow what your doctor says, but, you know, to take control of their own health in that respect. There is a fear, and obviously the more informed you get, the less fearful you are, right? And you can, and the reality is, the more involved you get in your own healthcare, the better your outcomes are going to be.
Yeah, I agree completely, and so many interesting points in there. You know, I'm all about working with health coaching clients with a doctor in their care as well. You know, it's about augmenting your care as much as you possibly can. And I think self-knowledge and knowledge about the processes involved is so important. You know, epigenetics is such a fascinating area. The expression of what genes you already have. That's sometimes why I think that giving genetic reasoning for example, obesity is kind of circular, because in a way everything is genetic, but it's how those genes are ultimately expressed. You know, like in Nessa Carey's book, The Epigenetic Revolution, she just uses the example of the Romeo and Juliet Shakespeare script. You know, you can watch the Richard Burton version, or you can watch the Baz Luhrmann version. It's the same script, but I mean, that's completely different what you're seeing. So similarly with genes, I think looking at the epigenetic expressions in different metabolic backgrounds is really super powerful. What do you think is emerging as the sort of most important things coming out of the research at the moment? And if it's too early, then what is it that people can do to help at the moment?
I think there's more of a focus on repurposed drugs which wasn't there before. I don't know if that's come about by pressure from certain organizations like the Care Oncology Clinic, or from patients, but that's definitely being discussed a lot more, and there's actual conferences on that from organizations like ABCAM, which is fantastic. But it tends to always focus on these pharmaceutical agents, and creating new ones rather than repurposing sometimes. So that's an interesting thing to note. It's rarely on diet, fasting, that kind of thing. But yeah, it's definitely creeping into, not creeping, but it's getting into the conversation more, which is a good thing. It's just, I found
let's not say creeping. Let's say it's confidently knocking on the door, and people are answering. Yeah,
it's being taken more seriously. And one thing that I became quite frustrated with, which is how this study came about, is since 2015 I've been trying to get involved with a ketogenic diet clinical trial, which has been in the discussion phase for many years. And I'm not going to say who's been involved with that, but it was just very frustrating. And so I thought, I'm going to do something myself, and it doesn't necessarily have to have any intervention at first. I just want to get the ball rolling. And so this is how this came about, just from a conversation with Isabella. And I've been impressed with the work that she's been doing. But yeah, overall, I think there is more of an interest in this area. It just needs more, I guess, more funding and more opportunity to do research that doesn't involve some big pharmaceutical company.
I think that's been the biggest thing, hasn't it? Is really sort of, it is a funding issue, because the reality is that most funding does go to drug trials. That's where it really goes to. It's either understanding more genetics, or it's drug trials, or the combination of the two. And you really don't get a lot of funding when it comes to lifestyle measures. And let's say where there might be funding with regards to it's a little bit, it might be interventional, but it often is epidemiological. I would say, might sound a bit controversial, but I would say it veers towards the low fat paradigm, and there hasn't been the testing of the low carbohydrate, high fat paradigm, not properly. And where there are touches on it, there's a mistake where there's only testing of urine ketones, maybe blood ketones, but only in the morning. And it's very misleading, actually, in that respect. And so, and there really isn't a sort of solid basis of research to establish that metabolic phenotype in cancer patients alone. Just, one particular type of cancer patients, like really clean, and obviously having the converse group to compare against. And it does require funding. People, people I know are always so generous to donate to charities, which is obviously the first place you go to, because one doesn't know really how to donate directly to a project. And what happens there is, the charities are looking for good projects, and those projects just happen to tend to end up in either pharmaceuticals or nutraceuticals. And so what we're doing essentially is, what is missing is that metabolic phenotyping. And is, as Andrew was saying, it turns out, because I am doing for my PhD research the control group, essentially. So it's sort of like serendipitous that we get that control group to compare against. And I wouldn't, not say serendipitous. I actually partly that was the purpose of my PhD, was, I said, I want my research to sort of be that foundation that we can compare all other hyperinsulinemia disease groups against, because that's what's necessary. Because this idea that you take sort of the normative population to compare against, well, how many of them really are actually unhealthy? They're actually in that hyperinsulinemia spectrum, really. And so that's why you get all this noise and you don't actually see that clearer picture. And that's why, you know, a lot of doctors will be like, no, there's not really any difference. We really actually need the two further ends of the spectrum. And so with regards to the research, that's and the funding, you know, obviously my PhD is separate, which is self-funded in actual fact. And what we're trying to actually capitalize on is, whilst I'm doing this, I have the setup, you know, the laboratory space, all the clinical work, all the consumables, the team, all these things. And that actually massively reduces the cost of the research, because there is a huge amount of cost behind research. You know, the team themselves, you know, they've got to live, they've got to survive, all these things. The consumables are ridiculously expensive. Lab space, you know, machinery, all these sort of things. So anyhow, I am doing that. So it makes so much sense right now to do that project and research for brain cancer patients, where we can essentially almost half the cost of that research because of the situation that we're in now. And it is really hard in terms of going to, let's say, a charity, going to them and saying, hey, look, can you fund this project? And, you know, they've got a gazillion people vying for funding, essentially. And so, you know, really on Andrew and I talking, we said, you know what, let's do it ourselves. I mean, I did it for my primary research, so we can do it again. Let's do it. Let's, you know, everyone else in the public there who says, we want to get this done. We're not going to wait for some, you know, fund or trust. We're going to do it. So let's all pull together and make it happen. And we can. So that's where we are now.
Yeah, I think Isabella and I are very similar. We don't like to wait for things to happen or just try to rely on others all the time and get continually frustrated. We just are people that like to get things done and do it ourselves if need be. So yeah, sometimes
Brilliant.
what's needed.
And what would the next level up look like, and when do you think that might happen?
You want to go, Andrew?
So I'd say after we've gone through 10 to 15 patients, it would likely be, I'd say, about a year and a half to get that completed. And then the next phase would be, I don't know, after we've seen what kind of patterns we can spot and what makes some meaning out of all of the data that we collect, because it will be quite a lot. quite a lot to go through.
I mean, essentially it's like this: if we were to go by hypothesis, so hypothesis is sort of, you know, us saying, well, this is what we see, this is what we think. I mean, we're doing this project because we think this, right, on the data that we have. So essentially metabolism is dysregulated. That seems to be on the spectrum of hyperinsulinemia. Where it sits on that spectrum, we're not 100% sure. It's probably varying depending on the stage of your cancer, type and stage. But essentially the argument is that there's a massive metabolic component to cancer and brain cancer. So then the plausible argument is, well, how do we reduce the hyperinsulinemia? How do we get that insulin into a healthy range? So the first part of that, in terms of dietary, so that's the really sort of large part that the patient can control, is to restrict the carbohydrates. And there's a fair amount of research that's showing getting into ketosis, these therapeutics have a positive effect. And so with the phenotyping that we're going to do, you know, based on hypothesis, assuming it shows the clear data in terms of being on the further end of hyperinsulinemia or just on that spectrum, the next stage would be to actually do an intervention and having that published work, to have the correct ethics application, to be given the ethics to do a randomized controlled trial. Although we've had discussions about randomization as opposed to actually doing a self-selecting study, because there is a lot of strong evidence that shows that when a patient selects the type of treatment that they do, they're actually more likely to adhere and have positive outcomes. So, you know, whether it be an RCT or not, or actually an open label self-selecting study, where there would be intervention of therapeutic levels of ketosis. And this is where it gets a lot more fine-tuned, because where there have been studies of ketogenic diets getting into ketosis for cancer patients, my own observation when reading those published literature, that I think there are areas that are flawed. And what they do is because they essentially try to control for the calories, and also often give the same kind of meal, if not a shake. And there are three factors involved in that I think make them flawed. The first is being in therapeutic levels of ketosis actually makes you spontaneously fast. And that's actually quite important. The fasting aspect is quite important. And spontaneous fasting has a different effect to obviously being calorically restricted and hungry and wanting to chew your nails off. Then the other aspect is controlling for the meals, because individuals not only have taste preferences, they have response differences to different types of foods. And so if I just give you the anecdotal evidence that I have friends who can eat broccolini who are in ketosis, no problem. Broccolini for me halves my ketones. It's considered a keto food, you know, and yet it really negatively affects me. I've had situations where I've seen some people drink kefir and have no problem whatsoever, and other people drink kefir and lose their ketones. So you can't be placing the same meal in front of people. And it quite frankly doesn't fit the real world situation. You know, at the end of the day, you're going to have people with cancers from different cultures who are going to want to eat different foods. And that's going to play a massive part on adherence to this kind of treatment. Then the other aspect is making ketosis shakes. They very much are usually full of vegetable oils, you know, and these vegetable oils are, you know, well indicated to be highly inflammatory. And when your body's in an inflammatory state already, I don't think it's a very good idea to be pouring a whole load of vegetable oils into your body. So, you know, these are three levels of what I think are actually quite flawed in ketogenic diets for interventions for cancer. And that's why you end up with a very low adherence to start with, intolerability, and actually not seeing the changes that you think you would see in terms of blood ketones, and then therefore disease progression or regression, because they're, you know, been controlling what people can eat in terms of, you know, therefore let's all give them the same plate, the same dish all the time, is not taking into account the individuality in terms of how they respond with regards to their ketones, insulin and glucose, as well as taste preferences. And so this is something that we want to massively avoid. So, you know, should we be successful and get to that next trial, when it comes to doing an intervention study, it will be not about giving people a controlled plate with a certain amount of calories with exactly the same dish between each of them. The intervention is getting into therapeutic ketosis. That's what it is. And, you know, there'd be a lot of help, a lot of coaching, really one-on-one. And then obviously assessing all of that and then putting that data together and presenting it.
It sounds great. I'm very much looking forward to seeing it happen. I know, Isabella, that you might have to duck out for a couple of minutes, but that you might join for the last couple of minutes. Yeah, that's fine. So Isabella's away. It's there's so much to go wrong, you know, just listening to Isabella there, and all the thoughts I've had about what is the ideal study for working out the best route for this type of thing. And it's great to hear Isabella talking about all the problems associated with the kind of utopian ideal of a keto diet. You know, people sometimes just aren't interested in doing it, you know. Modern foods are a kind of technology that, like any technology, you can't just put it back in, you can't put the genie back in the bottle. People love addictive foods that are terrible for them.
I think that's why we see so many keto carb replacement foods on the market, and some are okay because they have more natural ingredients, and others are just full of things that full of ingredients that are going to at best disrupt the gut microbiome, and at worst not even put you in therapeutic ketosis an hour after eating. So yeah, there's many problems with, especially if you're on a calorie controlled ketogenic diet, to be able to metabolize the nutrients that you're eating when you're having all these convenience keto foods. That's one of the problems I noticed just since, well, when I was diagnosed, none of these foods really existed, so it's kind of easy for me. Another issue that we may have in that intervention stage is there's often, or we have mostly to go on is ketogenic diets for epilepsy, and for cancer, that's likely to be a bit different. So that's a consideration as well.
Yes, I was thinking about how there's probably a route into some kind of funding from ketone manufacturers ultimately for certain things, but you would think that for people in your phenotype 2 or 3, if they're just chugging ketones, then cancer's probably not the
well, if they're ketone esters, that could be a different story.
Well, potentially, yeah. I mean, you wonder whether still eating all the stuff that you're used to eating that got you into phenotype 2 and 3 in the first place, and ketones. If you're epileptic, if you're bipolar, well, maybe all you need is high ketones, and but it might not be the same for all pathological problems, right?
Yeah.
Well, yeah, I mean, if you're assuming that the person who ended up with a disease, a metabolic hyperinsulinemia disease, let's just assume they were eating ice cream and pizza, or even like, forget that, let's not do like really, really like the most obviously terrible things, but they were eating, let's say, porridge for breakfast, and some sort of lentils at lunchtime, and what would be
heart healthy salad dressing.
Yeah, and so that's heart healthy salad dressing is using canola oil or sunflower oil, things like that.
Yeah, with agave nectar, so that it's not refined sugar, all that malarkey.
Yeah, or honey, lots and lots of honey, or they're going to the gym lots and they're having banana shakes with some peanut butter to get the protein and glucose up for protein synthesis, muscle synthesis. So looking at those sort of things, these things are going to affect inflammatory pathways, and also the fatty acid profile that they're getting from that diet is going to affect the type of fats in the membranes of their cells, and that's going to consequently affect the ability for things like glucose receptors or insulin receptors to be on those membranes and to be able to conformationally change. So the membranes on your cells are not fixed, they're not solid. It's called a fluid matrix, and so all the lipids are moving around, and there are these little rafts in these lipids that are usually composed a lot of cholesterol, and the fluidity of this membrane is affected by temperature and affected by the type of fatty acids and cholesterol components inside. And so the more vegetable oils that you have, seed oils, things that are historically were not available to us until modern manufacturing, really, they're going to be changing the fluidity of the cell membrane. And that fluidity is then going to affect the receptors that are plugged in, first, to be able to be plugged in. And secondly, a receptor has to be able to move, has to be able to open and shut, has to be able to change its shape to be triggered or to allow its substrate in or out of the cell. So those factors are not even to do with triggering insulin release or excess glucose. It's affecting the membrane health of your cells. So the quality of ingredients in your food is going to have a profound effect on your cellular health and consequently tissue and organ health just there. But if you're talking about having, the same diet, not making any real change, but having higher level ketones, there was a really great paper, and it was studying type 2 diabetics who were put on sodium glucose transporter inhibitors. So it's basically a medication that helps you pee out glucose more. And one of the side effects of this drug is that you can actually end up with high levels of ketones with high glucose. And that usually rings alarm bells. And it's often called ketoacidosis. Now, real ketoacidosis is not just pathologically high levels of glucose with high levels of ketones. It's also with low levels of bicarbonate, which is a buffer for the acidity. So really it's an under 22 level marker of bicarb. But anyhow, it's commonly referred to as diabetic ketoacidosis. And where they found that there was a sort of low-grade diabetic ketoacidosis, so they had high glucose levels because they're diabetic, they're uncontrolled diabetic, so they have to be on drugs, and they sort of have blood glucose levels of 6 and 7, but they're also having higher levels. They're having presence of ketones essentially with these high glucose. They actually found that it was cardioprotective, that these patients actually suffered less cardiovascular events as a result. So it's a very, very interesting finding. So this is actually also showing you essentially an independent aspect of ketones being a positive effect.
And for those people, possibly those numbers of 6 and 7 millimolars of the blood glucose might not necessarily be high for them.
This is very true too. Yeah, because it's all about reference ranges, right? So you can have those. And I have to say, I have seen that in long-standing healthy ketosis individuals where they've had, 5.7, 5.8, ranging nearly into 6 with their glucose, with really good ketone numbers too, and having tested all their blood markers, like not all, but more than 60, being in superb health. Absolutely superb health.
I actually I had that when I was playing badminton last week on Friday. So I played badminton for the first time in over 10 years. And I just curiously, out of curiosity, in the middle of playing, I tested my blood glucose and blood ketones. And my blood glucose was a bit higher than normal, so it was around the 6, 7 range. But my ketones were still in the therapeutic range for me. It was between 3 and 5. So I just found that interesting because exercise obviously brings up your blood sugar. And exercise for me, if it's glycolytic kind of activity, can be a seizure trigger. But when my ketones are high and I'm exercising, I have a higher seizure threshold, so it doesn't affect me. But yeah, that was interesting. That's kind of
like argument for sort of evolutionary events, right? I mean, if you assume, like, first of all, stress is going to elevate your adrenaline, and it can therefore elevate your blood glucose. And
facing a tiger.
Yeah, facing a tiger, facing some sort of imminent threat, your body wants to elevate the blood glucose for a number of reasons. The first would be making sure it's available for your heart and your muscles so you can run when you need to. Another reason is, let's say you are injured, you fall over, you cut yourself, or the tiger gets you for a bit and bites you somewhere. You want to be able to coagulate so that you don't bleed out. So these are incredibly reasonable. And at the same time, if your background is always to be in ketosis, then first of all, you wouldn't want to actually have your ketones go down in that event either, because if your muscle cells and your heart is guzzling up that extra glucose, then you want to make sure there's enough ketones for the brain so the brain is well taken care of in that time. But also in terms of the ketones being there to counter any of the negative effects of being put into a hypercoagulable state. Should you have a cut, an injury in that sort of event, then you'd want resolution. You'd want healthy resolution. So having that high level of ketones there, arguably is what enables that healthy resolution there as well, right? So
yeah, I mean, I love looking at any of these questions through the lens of our evolution, and to sort of bring it back full circle to where you started, Isabella, talking about how we evolved. When we look over the last few hundred years at populations who were in phenotype 1, they didn't have the diseases of modern civilization. And although it's not just eating that gets us there, I've talked to Andrew about this and how there's hotspots of cancer where there's particular environmental pollutants and stuff like that. It's not just an individual thing; it's a phenotypical thing, and it's great when people like yourselves start illuminating the recesses of darkness that are still so prevalent. So I really appreciate you coming on. I think I would like to keep talking for ages, but I know that you've got another meeting to go to. So if you've got anything else that you want to say, then please go for it, and let people know where they can find you and how people can help with the research.
Well, we welcome additional funding. It's all helpful. So we do have a website. It's www.beatbraincancer.co.uk. It's a catchy title. We did have another title, but it was a bit more wordy. So yeah, beatbraincancer.co.uk. And also a Twitter account. If you follow Cancer Met Study, there's some interesting research that we like to tweet out, and when we're actually starting, we'll be tweeting updates so people can keep up to date there. And I think that's about it, unless Isabella has anything else she'd like to add.
That's it.
Well, I really appreciate you both coming on again, and more power to you both. There's a lot of things that any of us could do with our limited time in this life, and I appreciate that you're both doing something that's ultimately going to help a lot of people.
Yeah.
Thanks, and thanks for everything you do, Ali, as well.
Oh, you're very welcome.
Thank you for having us. Thank you.
Hey everyone. This episode is going out on Wednesday, the 11th of October, 2023. And I just want to talk about our small group sessions, coaching sessions, which are starting again on this Monday, the 16th of October, 2023. It's 8 90-minute sessions running Mondays at 7 p.m. UK time, worldwide on Zoom. Going right through to Monday, the 4th of December this year. So what do you get in the small group sessions? You get cutting edge science explained clearly and in simple terms. You get discussion of subjects that affect your metabolic mental health, including food addiction, dietary modes like keto and carnivore, why it works, how to get your macros right, gut health, getting enough fat, supplements. And you also get a non-judgmental and totally confidential space to talk about what matters for your mental health. With experienced practitioners, myself and senior psychiatrist, Dr. Rachel Brown, who understand how powerful diet and lifestyle can be. So what have other people said about doing this small group session with us? Well, Martina said, great, it's geared at just where I was at and just what I wanted to know. Ali and Rachel's knowledge and commitment to metabolic psychiatry and their desire to share this was an absolute gift. Elizabeth said, go for it. I can't believe the difference it made. I still have more to problem solve, and it's great to still have the forum and possible subscription, but finding freedom from binging has been a dream come true, and many times where the clouds of depression parted has been so good. And Chris said, I would actually say this is an exciting and cutting edge therapy. It's something I wish I'd known about 30 years ago. We won't be recruiting again for small group coaching until February 2024. So if you've wanted to work with myself and Dr. Rachel Brown and you feel like the group sessions are the way to do it, you have to act now, because they start on Monday at 7 p.m. UK time, worldwide on Zoom. In previous groups, we've had people from America and elsewhere. So if you can make 7 p.m. every UK time, which is about midday to 2 p.m. ish on a Monday in America, every Monday from next Monday, the 16th of October, until Monday, the 4th of December, and just go into Christmas time feeling great. Then head over to bit.ly/MetSciGroup, METPSYGROUP. I'll put it in the show notes as well. If you're on the fence about it and you're not sure about the process, then you can have a free 30-minute chat with me, either about the group sessions or going one-to-one, and that's at bit.ly/IMETPSYGROUP. Ally Transforms, ALLY Transforms. I'll put that below in the show notes too. So please head over to bit.ly/metasci group, book yourself a place. There's 20% off now, and it's a great deal. So please enjoy today's episode with Andrew and with Isabella, and I'll see you soon.